Today we'll talk about the Mendels hybridization experiments and what are the pure lines. Mendel was curious, he was interested to know how traits are passed from parents to offsprings. And to determine how traits are transmitted from parents to offsprings, Mendel performed hybridization experiments on the pea plant Pisum sativa. Mendel performed his hybridization experiments in 3 stages. Mendel performed his hybridization experiments in 3 stages. Here we will talk about the Mendel's hybridization experiment in pure lines. I told you that this Mendel, he performed his hybridization experiments in 3 stages. First was selection of pure lines, or true breeding plants. Selection of pure lines or true breeding plants. Second is cross between pure lines, to obtain F1 generation. Then first selection of pure lines, then cross between pure lines to obtain the F1 generation. Third is selfing of F1 plants and raising F2 and F3 generations. So this was the steps which Mendel followed during his hybridization experiments. That means selection of pure lines or true breeding plants, then cross between these pure lines to obtain the F1 generation, what we call as hybrids. Then helping of these F1 plants to raise the subsequent generation like F2 generation and F3 generation. So this now the question arises that how these pure lines are produced. These pure lines they are produced by means of self-pollination, and self-fertilization leads to development of pure lines. Self-pollination and self-fertilization. Leads to development of pure lines. And through many generations of natural self-pollination and self-fertilization, this Mendel developed the pea plants into the pure lines. So what are the pure lines? How we can define the pure lines? Pure lines are true breeding plants. They are pure lines. Or what are pure lines? Pure lines are true breeding plants. That is, they produce the offsprings which are phenotypically identical to their parents. That means the offsprings which are produced by these pure lines, they are phenotypically identical to their parents. So these are called as pure lines. So pure lines are true breeding plants. They produce offsprings which are phenotypically identical to their parents. That means pure lines are homozygous. Pure lines are homozygous. That is, they have two identical alleles for the trait. They have two identical alleles for the trait. For example, if we'll say that capital T for tallness, small t, these are pure lines. So they have two identical alleles for the gene. And true generations, they produce the same trait generation after generation. Another definition is pure lines. They produce same trait generation after generation. So what are pure lines? Pure lines are true breeding plants. That is, they produce the offsprings. These offsprings, they are phenotypically identical to their parents. Or we can also say that pure lines are homozygous for a gene, for a character, for a trait. That means they have two identical alleles for the gene. They have two similar alleles for the gene. Or true breeding plants, what are called as pure lines, they produce the same trait generation after generation. For example, if we have got a pea plant with round seeds, if a two pea plants, this is a pea plant with round seeds, and this is a pea plant with round seeds. That means this is pure line, this is pure line. Now when they are crossed with each other, their offspring is a pea plant which has round seeds. So this is what is called as pure line. That means the offsprings which are produced by pure lines, they are phenotypically identical to their parents for that particular specific trait. So then this Mendel, Mendel took 2 years for checking that his hybridization, he took 2 years for checking that his hybridization stuff, or we can say that experimental plant, pea plant, is true breeding or pure line. How he confirmed? He confirmed that the pea plants which developed from the round seeds, they would produce the offsprings which have round seeds when they were crossed with each other. For example, if this is a pea plant with round seed, and this pea plant is crossed with another pea plant with round seed, then they will produce the pea plant with round seed. That means this is pure line. Pure line. They will produce the offspring which is phenotypically identical to their parents. This is what is called as pure line. Then this Mendel, he experimented he performed his experiments on the pea plant, and this pea plants on a different varieties of pea plants, and these pea plants, they differ in 7 contrasting traits. So he crossed these pea plants one by one. For example, how he performed his experiments. So Mendel crossed pure line. Mendel crossed two pure line pea plants which differed with by one trait. For example, seed shape, for example, height. So Mendel crossed two pea plants. He crossed two pea plants, but these two pea plants are pure lines and they differed in one trait. For example, seed shape, seed color, pod shape, pod color, या height, flower position, या flower color. For example, this is parental generation. This is pea plant with round seeds. This is pure line. So pure line, it has two identical alleles for a gene, and it was crossed with pea plant with wrinkled seeds. This is also pure line. This is smaller, and in the F1 generation he obtained what? This is pea plant with round seeds. This is pea plant with wrinkled seeds. So F1 generation he obtained pea plant with round seeds. So pea plant with round seeds. So this is hybrid. This is hybrid. It is called as F1 generation. So these and in the next generation, when this F1 plant were this selfing, he obtained pea plant with plant. In the F2 generation he obtained pea plant with round seeds and pea plant with wrinkled seed. So it was like this, or and the genotype was smaller. So we can say that how this Mendel performed his hybridization experiments. He used pure lines for his hybridization experiments, and Mendel performed his hybridization experiments on varieties of pea plants, and these pea plants differed in 7 contrast pea plant varieties, I can say, which were these pea plant varieties which were growing in his monastery garden. They differed in 7 contrasting traits. For example, stem height. So he took into consideration the stem height of the pea plant varieties. Seed shape. He took into consideration the seed shape of pea plant varieties. Seed color of pea plants. Pod shape, pod color, flower position, and flower color. These are 7 contrasting traits which were selected by the pea plant for his hybridization experiments. And we know that this stem height, it has two forms, it has two contrasting forms. That means it has two contrasting forms. That means some pea plant varieties were tall and some were dwarf. Same is the case, this seed shape, he crossed the pea plants which are tall with the pea plants which are dwarf. Same is the case, this seed shape, it is having two contrasting characters. This trait, it is having two contrasting forms or two contrasting characters. This is round, seed shape can be round and wrinkled. That means there are some pea plant varieties, pea plants which are having round seeds, and some pea plants are having wrinkled seeds. He crossed the pea plant with round seeds with the pea plant with wrinkled seeds. Same is the case with seed color. Some pea plants were having seeds which are yellow, and some pea plants are having seeds which are green. So he crossed pea plants, and he crossed pea plants with yellow seeds with the pea plants with green seeds. And in the F1 generation, he obtained yellow colored seeds, pea plants with yellow colored seeds. Then in this pod shape, some pea plants are having inflated pod shape, some are having constricted pod shape. Then what he did, he crossed the pea plants with inflated pod shape with the pea plants with the constricted pod shape, and then evaluated the results in the F1 generation. Then pod color, some pea plants are having pod color green and some pea plants are having pod color yellow. He crossed the pea plants having pod color green with the pea plants having pod color yellow, and then in the F1 generation he obtained the pea plants with green pod color. In this way he observed that how traits are transmitted from parents to offsprings. And in the same is the case, in his monastery garden, some pea plants were having flower at the axial position, in the axil of the leaf, axial position, and some pea plants are having flowers at the terminal position. And what he did, he for example, this is a pea plant with flowers in the axial position. He crossed it with the pea plant. For example, this is another pea plant with the pea plant having flowers at the terminal position. And then in the F1 generation he obtained pea plants with flowers at the axial position, because axial position is dominant over this axial position is dominant over the terminal position. Same is the case with the flower. This flower color, it is also having two contrasting forms, two contrasting characters. One is purple, what is called as colored flowers, and another is white flowers. So he crossed pea plants with purple colored flowers with the pea plant with white flowers. And in the F1 generation, what he obtained pea plant with purple or colored flowers, because purple flowers or colored flowers are dominant over the white flowers. In this way, Mendel performed his hybridization experiments, and he raised F1 generation and F2 generation. Then he performed his laws, different types of laws, Mendel's laws of what is simply called as Mendel's laws of inheritance. We have law of dominance, we have law of segregation, we have law of independent assortment. This was how Mendel selected his pure lines, then performed his hybridization experiments. Thanks.